SEngine was founded with the idea to prepare for the next generation of cancer treatments, where treatments are going to be targeted not just to a specific gene, but also targeted to an individual. We named our test Paris in analogy with Paris, who defeated Achilles by precisely aiming at its only weakness. Similarly, for cancer cells, we have no time to try different treatment for every patient. We want to aim right away to the right treatment for that patient. And that means to target the vulnerability of that particular patient cancer.
Most precision medicine works off of genomics data alone to give clinical recommendations to their oncologists. SEngine offers a CLIA-certified functional chemosensitivity test for their clinical recommendations in 4 to 6 weeks. SEngine is the only CLIA-certified company that offers this test, this functional assay test for solid tumors.
At SEngine, we are using the patient-derived organoid cells and the technology of the echo to test the new drug candidates on cells directly derived from patient samples. And that really allows us, instead of finding a drug which is really good in killing one cell line, we are finding drugs which are targeting and helpful for a cancer type or with a certain biomarker, instead of just focusing on a single cell line.
Our goal is to have the best system in place to optimize those organoids, grow them in culture, and get them screened as soon as possible. As soon as we have clear, distinct organoids, which are nice 3D structures, plate them in chambers and treat them with over 120 different drug combinations. Results are then converted into something that physicians and patients can understand rather than just raw numbers. And that will tell us with very high accuracy what drugs will most likely be effective in patients, and more importantly, which drugs will not be effective in patients. So not only are we likely to tell a patient what they will respond to, but what drugs they should not go on, even if it is the standard of care.
We've had patients who have come in who have been refractory to prior therapies, and we've screened those cells in combination with the drugs that they've become refractory to, and we have actually resensitized those cells to previous drugs. This gives these patients a whole new opportunity to assess drugs that they can actually receive in the clinic. One of my main responsibilities here is to make sure that our test is always cutting edge and always as good as it can be. The speed and precision of the echo has really cemented its place as the workhorse in our laboratory. Using the echo and the access system has enabled us to greatly increase our throughput from doing one plate at a time to queuing up dozens. We can create custom panels and hyper-personalize this test by making bespoke panels of drug combinations for every patient. The software suite available from LabSite really enabled us to design multiple dose responses and combination therapies and integrate them into our workflow very seamlessly. The ability to move around different drugs and actually apply the same drugs over different wells and arrange those combinations on the fly really enables us to interrogate these pathways in a whole new way. I think using the echo is going to enable us to finally approach functional medicine the way it needs to be treated.
With the echo, we can now, at will and in a custom fashion for that particular patient, test multiple drug combinations and really have a chance of recommending a treatment that might lead to a cure.